Dear student, Model 12, Numerical Chromosomal Aberrations, under that haploidy and polyploidy with suitable human examples. The main objectives of this episode are to explain about the haploidy conditions and their types, to study the evolutionary properties of haploids, to understand about the polyploid conditions, and to know about the significance of polyploidy conditions. Introduction. Tremendous diversity exists in organisms related to their ploidy level. That is, the number of sets of homologous chromosomes per cell. This change, the ploidy, represents a significant genomic difference among the organism, and it mainly affects the evolution of species. Because of this, it might also be thought that the ploidy evolution would be well understood. When the study on ploidy evolution was done, it was assumed that how the evolutionary forces act on ploidy levels and they reveal additional levels of complexity. All sexual organisms have a life cycle that alternate between two ploidy levels. They consist of a reduced state that follows meiosis and a duplicate state that follows the union of gametes. Most of the organism, the alteration is between the haploid phase and the diploid phase. The haploid phase consists of one chromosomal set and the diploid phase comprised of two chromosomal sets. In the case of animals, the haploid phase is restricted to only unicellular gametes which do not undergo further mitosis. This is followed by the gamete fusion which leads to the diploid phase. This occupies the entire lifespan of the organism essentially. It is also important to note that some animals consist of haploid males who are produced parthenogenetically, which is the reproduction of individual from the egg without fertilization and the diploid female production by sexual reproduction called archaeotoky. In the case of archaeotoky parthenogenesis, the unfertilized eggs developed into males. Archaeotoky occurs in members of Hymenoptera including bees, ants and wasps. It can also be seen in some spider, mites, bark beetles and rotifers. In the case of plants, mitosis occurs in both haploid and diploid phases. The diploid phase is predominating in some ferns and seed plants. Apart from this, the polyploidy condition is very common among plant species. Having more than two sets of chromosomes become evident that all flowering plants have polyploidy history. In most of the cases, the polyploidization has occurred very early in evolution, where many of the duplicated genes have diverged substantially or been lost. Hence, in this episode, the following aspects are going discussed. A. Haploidy condition and their types. B. Haploidy in mammals. C. Polyploidy condition and their types. D. Polyploidy in human cells. And E. Significance of polyploidy conditions. A. Haploidy conditions and their types. The haploid condition shows the presence of only one set of chromosome. If a cell contains half the usual number of chromosomes, it is termed as a haploid cell. In the case of eukaryotes, the cells normally consist of diploid number of chromosomes. In gamete cells, the meiosis occurs and the number of chromosomes gets halved. Depending on some cytological features, the haploids are classified into haploids and aneuploids. Euhaploid is the condition in which the number of chromosomes is an exact multiples of one of the basic number of the group. Euhaploids are further classified into monoploid or monohaploid and polyploid. Monoploid arises from the diploid species and they consist of basic chromosomal number of the species. The polyploid is the haploid individuals who arise from the polyploidy species. They can be dihaploids, trihaploids and so on. The individuals having chromosome number which are not an exact multiples of the basic number of the group. This can be further classified into disomic haploids n plus 1 having one extra chromosome belonging to the genome, additional haploids n plus alien chromosome having few other chromosomes which are not belonging to the genome of the individual involved, nullisomic haploids n minus 1 with one chromosome missing, substitution haploids n minus 1 plus 1 alien, where one or more chromosomes of haploid individual are substituted by alien chromosomes, and misdivision haploids consisting telomeric or isomeric chromosomes that results from the misdivision of centromeres. Evolutionary properties. Organisms with different ploidy levels exhibit different types of evolutionary properties. The efficiency of the selection and the number of mutations are the two important keys that differ in the evolutionary process of haploids and diploids. These evolutionary forces act on organism and shape the ploidy evolution to remain diverse. The number of mutations that occurs in individuals is directly related to the ploidy levels. Diploids having double the number of chromosomes can show a number of mutations than haploid. In some cases, having more mutation is selectively advantageous, depends on the fitness of the organism. It also depends on the rate at which its environment changes. If the organism gets adapted to the environment correctly, mutations only serve to push the individual further away from its fitness to that environment. Haploid with less deleterious mutations gets favored in those situations. If the organism is not adapted well to the environment, or if the environment gives more challenges of variation, diploids may get advantages and gain the upper hand by producing more mutations. The evolution of haploids or diploids does not depend only on the number of mutations, but also on the fate of these mutations and the selective effects. B. Haploidy in mammals. The condition of diploidy can easily get deviated in the case of tumorigenesis. The genetic instability and chromosomal aberrations are the important hallmarks in case of cancer. The tumor cell can show condition of aneuploidy, having an irregular number of chromosomes because of the gain or loss of chromosomes. Polyploidy condition can be seen if the whole genome gets duplicated. In very rare cases, the chromosomal loss can be observed, which results in nearly haploidy conditions. These conditions are found in leukemic human cell line KBM7. These cells are transformed and harbor additional genomic alteration, including some translocations, that is BCR to ABL1 translocations. Usually, the germ cells, the oocyte and sperms are haploid as a consequence of reductive division in meiosis. Parthenogenetic embryo formations can take place that carry exclusively maternal genome without the paternal genome. In the case of the parthenogenetic embryo, the haploid oocyte can begin cleavage and early embryonic development without fertilization. The parthenogenesis can be seen spontaneously in woman, but the resulting embryo cannot survive. In other cases, woman parthenogenetic cells can divide continuously and generate benign ovarian tumors termed as teratomas. The diploid ovarian teratomas can be formed from a haploid oocyte, and they are completely homozygous. The haploid oocyte can also undergo endo reduplications, that is the formation of elevated nuclear genome content, or polyploidy by the replication of the nuclear genome in the absence of mitosis or cell division in later stages. The parthenogenesis can also be induced to oocyte cell by chemical or electrical exposure, which mediates the activation that mimics the fertilization. Polyploidy condition and their types. Polyploidy is the condition in which the presence of one or more additional sets of chromosomes instead of a normal diploid condition. A diploid organism consists of cells with usually 2 sets of haploid chromosome number. In case of polyploidy condition, the presence of 3 or more haploid chromosome sets can be observed. The whole set of chromosomes fails to separate during meiosis or mitosis, result in organism or cells with polyploid condition. Usually, the polyploidy conditions can be arise due to the non-disjunction during mitosis or meiosis. Polyploidy conditions are usually common in flowering plants and less common in case of animals. Some of the invertebrates, frogs and lizards can show the polyploidy conditions. Polyploidy condition can occur in some of the tissues in humans, including liver cells, heart muscle cells, bone marrow cells, which are highly differentiated cells. Polyploidy can also be seen commonly in amphibians. Polyploidy can be classified into autopolyploidy and allopolyploidy. Autopolyploidy is also referred to as autoploids. They consist of basic chromosome sets that contain multiple copies of the same genome. Autoploids can be formed from the improper cell division or through the union of unreduced gametes in nature. They can also get induced artificially. Some natural autoploids include tetraploid plants such as peanut, 4n equals 40, potato 4n equals 48, triploid crop like a banana 3n equals 33, and hexaploid sweet potato 6n equals 90, etc. Naturally, they can occur spontaneously by chromosome doubling, which has a varying effect based on the species. Spontaneous doubling of chromosomes in case of ornamental plants such as tulip and hyacinth, etc., and forage grasses such as ray grasses has led to increased vigor and yields superior varieties. By the process of induced polyploidy, breeders can produce superior crops. The induced autotetraploid in case of watermelon crop produce seedless fruit with increased size. In the process of induced polyploidy, the diploids are treated with some mitotic inhibitors such as colchicine and dinitroanyls. To check the results of induced polyploidy, some approaches including the chloroplast count in gourd cells, checking of morphological features such as the size of the flower, leaf or pollen, etc., can be done. Allopolyploidies are also referred to as alloploids. They are formed from the combination of the genome from two or more other species. Hence, the chromosomal composition of alloploids is derived from two different species. The hybridization of two or more genome followed by chromosome doubling or fusion of genomes between the species can result in the allopolyploid conditions. In the case of angiosperms and ferns, these processes lead to the speciation. Some of the economically important alloploids include strawberry, wheat, oat, cotton, oilseed, rape, blueberry and mustards, etc. Karpachenko performed some classic experiments that started research on the alloploids in 1928. In one of his experiments, he knew that the cabbage and radish both had a diploid number of 18 chromosomes, and he had a thought of getting offsprings with 18 chromosomes. His goal was to get a new plant with radish root and cabbage heads, but unfortunately the progeny formed from the cause appeared sterile. It gave a conclusion that correct pairing of chromosomes and the normal function was not possible. Hence, all the gametes formed become non-functional. But surprisingly, he observed the appearance of some seeds. They were grown later and chromosomal analysis had revealed that their diploid number was 36. This suggested that the chromosomal doubling had occurred. D. Polyploidy in human cells. Polyploidization is not common in normal human somatic cells. Some variation at the ploidy level can be observed in different body cells. In case of polyploidization, the duplication or fusion of entire genome can be occurring, unlike the duplication of a single chromosome, which normally leads to the aneuploid condition. The extraembryonic part of the placenta consists of giant cells that can form multiple cell types during the embryonic development to form the adult body. These cells are polyploids. There are also other polyploidy cells in the human body, including hepatocytes or the liver cells, myocytes or the muscle cells, Purkinje cells in the brain and memory cells in mammary glands. The proportion of polyploidy can increase with the aging process or with other cellular stress, including surgical research, toxic stimulation, metabolic overload or some other oxidative damages, etc. Hepatocytes polyploidization in the liver is generally considered as the indicator for the terminal differentiation, that is, losing the ability of proliferations and course of acquiring specialized function, cellular senescence, and also related to the dysfunction of insulin and p53 pathways. Besides the mesothelium and the urothelium acting as the membrane lining, thoracic cavities and urinary tract respectively can also be in polyploidy conditions. The mechanism of formation of polyploidy can vary across the cell. In the case of muscle cells, the polyploidy can occur through fusion of genome, whereas in another cell called megakaryocyte, the polyploidy can drive by the repeated endo reduplication. E. Significance of ploidy conditions. The multi-allelic nature of the loci in case of polyploids becomes the main advantage that is very useful in breeding. Also, high frequencies of chromosome mutations are used in modern breeding techniques, including tilling, where they provide variations as new sources. The concept of gene redundancy and mutation tolerance in polyploids increases crop improvement. Production of seedless fruits. The seedless trait of triploid plants has become a desirable trait, especially in case of fruits. Triploids are produced artificially by crossing tetraploid with the diploids. The commercial use of these triploid fruits can be found in some crops, including watermelons. Some of the industrial application of polyploidy include the production of sex hormones and corticosteroids by artificial induction of tetraploids from the diploid plant. By the process of chromosome doubling in Chrysanthemum species, attempts are made to produce a botanical insecticide called pyrethrin. The production of secondary metabolites such as alkaloids, terpenes, often resistance to pests and pathogens. To conclude, the condition of polyploidy has many important roles in case of plants than animals, and it has been shown many industrial application in current days. The condition of polyploidy can occur more in nature than haploidy. Thank you.